ISIS is an interface that allows analysts to use a complex modeling framework to model the spread of infectious diseases through large realistic populations. ISIS provides enhanced workflow and reusability experience to end users. It allows users to design experiments to evaluate the effectiveness of pharmaceutical and public policy interventions, such as vaccination and school closure, individually or in combinations. Let's enter into the world of all new ISIS. The Experiments page summarizes the current list of experiments. When an experiment is selected, the user is able to view the parameters of the experiment, including the disease to be modeled along with the interventions and how infected people are introduced, which is referred as initial conditions. Now we will look at creating the new experiment. User can fill in basic details about experiment, including name, description, and selecting the simulation engine. Multiple replicates are run in order to bound the inherent variability of the system. The total number of simulated days can also be set, after which the data is saved and the user then proceeds to the next steps. Now the user can select the region that will be modeled. Next step is to select the disease model. Here user can define a new model or select an existing one. The transmissibility of a disease is the likelihood that the disease is passed from an infectious person to a susceptible person. The analyst specifies the probability that an infected person will spend a particular number of days incubating. The infectious period is the time during which an infectious person can pass the disease to a susceptible person. The next step is to define the way in which infected people are introduced into the system, referred to as the initial conditions. There are 3 different types of initial conditions. On day 0 specifies a number of people to be infected at the beginning of the simulation. Every day specifies a number of people to be infected at the start of each simulated day. This is meant to simulate the effects of infections that are brought into the simulated regions by people traveling from other infected regions. Daily seed specifies the specific number of people infected on the given simulated days. User can select either option. The next step is to define the interventions that are applied during the simulation. User can define 6 different types of interventions. Vaccination defines a vaccination strategy for a subpopulation. Social distancing reduces unnecessary activities, such as shopping or going to the movies. The close work intervention closes some workplaces, while closed school represents closure of schools. Antiviral treatment treats people who are sick with an antiviral drug, such as Tamiflu. Antiviral prophylaxis applies antivirals to an entire subpopulation, such as healthcare workers. The experiment definition is complete now and can be run. Once the experiment is completed, it can be analyzed. The analysis screen summarizes all the created analysis, along with their category, group type, and plot type. User can examine existing analysis, look at results, or create a new analysis. In analysis details, user can define basic information of analysis like name, description, and the title of the plot. 3 types of analysis categories are available. First is AP curves, second is reproductive number, and third option is TIGS, the tool for isolation group sizing. The user can select any completed experiment. For a selected experiment, user can select the cells to analyze. Now that the analysis is created, it can be run. Once the analysis is complete, the user can see the output by clicking on the View Results button. Plot shows the number of people infected on each day. The intervention summary shows details of intervention, applied. The means table and daily mean infection table provide numerical data and can be used for further analysis. Hope this demonstration has given you basics of ICIS application. Thank you.